one needs to be set up?
Nothing.
Oh.
No, the clips are already.
Alright, yeah, students are coming over already. So where's the best spot for us to sit?
Probably like in this row right here.
Welcome.
Thank you.
There's a
We did. We did. Flat bike tires, guys.
Sounds
From
like a morning.
is not fun.
You know, yeah, it's probably just because it was you.
It was Stephanie.
My bike.
But I mean, if you weren't with her, maybe you wouldn't have gotten Alright, we got papers for y'all.
Thank you.
Apparently we got a little thing we don't have to be fossil phobic and like open and close.
So, no, it's like, think of it like a 4-3-2-1 kind of thing. They all need to sit in the first 3 rows. Good morning. Front foremost.
That's not bad form.
You should have chosen the other tie. That's fairly blue, buddy.
Don't want to be blue. I have fun out of it.
It's like teal.
Yeah, I guess it's fun.
A little bit. I think that like right as I walked up a little bit, as soon as I started talking. Yes. Yes.
Is it something you want to pursue?
Yes. Right.
Well, I guess I'll find out. I have and this is the process.
So great. So you know, so you have to take it next semester. But it was all worth it. She retired. Yeah.
No, everyone sits in the front four rows. Everyone sits in the front four rows.
Very good. You want to go, maybe we can go, you want to go roll back? Greg, you want to go roll back now? Right? Are we going this way? I'm just going inside here. Okay.
Perfect. Hello.
that make sense?
And right here. The X is for Thursday. It's so you can see the screen and so that you are the posters so that you actually show up in the video. Because otherwise you get lost on the Yeah, so this is where you should all stand. Yep, just like this. That's for groups of two. Yeah. All right, ladies and gentlemen, thank you for coming. It's almost 9 o'clock, so we're going to go ahead and get started. Thank you all for coming. This morning we have Catherine Jacobs and Kate Blanchard as our first presenters.
Hi, my name is Kate Blanchard.
And my name is Catherine Jacobs, and we study tyrosine sulfation.
We worked under the supervision of Dr. Grace Rosenquist in the UC Davis Department of Neurobiology, Physiology, and Behavior.
And we are presenting for the Young Scholars Program 2019. So what is tyrosine sulfation? Tyrosine sulfation is a post-translational modification that enhances protein-to-protein interactions and signaling.
So the way it works is there is a sulfate ion that is transferred via the TPST catalyst, which is called tyrosyl protein sulfotransferase, but we'll call it TPST. And then the sulfate ion binds the hydroxyl group of the amino acid tyrosine, and this is the irreversible change that ultimately enhances protein-to-protein interaction.
So tyrosine sulfation was first discovered in 1954 by Bettelheim in bovine fibrinopeptide.
About 30 years later, Huebner discovered that it happens with the TPST catalyst, and he named it.
In 1987, Huebner discovered that tyrosine sulfation occurs in the trans-Golgi complex because that is where the catalyst TPST is located.
So throughout the 50 years after it was discovered, over 50 tyrosine sulfated sites were discovered. However, the scientific community still was not really aware that this was a chemical reaction that occurred, until finally in 1999. It was discovered that the chemokine receptor 5, or the HIV-1 co-receptor, is actually tyrosine sulfated.
So this HIV-1 co-receptor is also chemokine receptor 5, which is a G protein coupled receptor, or GPCR, and it has been later theorized that most, if not all, chemokine receptors are enhanced by tyrosine sulfation. So today, we know that 1% of all tyrosines are sulfated because Huebner discovered that 1% of all tyrosines in Drosophila fruit fly are sulfated. So this means that we have many tyrosine sulfation sites to find in humans.
So what we did is we are making predictions about which tyrosines are sulfated in different proteins in order to help the scientific community focus their research on specific residues, saving both time and money.
So now we will talk about our methods.
So first to define what a tyrosine sulfation site is. It is the amino acid sequence surrounding the tyrosine with the 5 amino acids before the tyrosine and the 5 amino acids after the tyrosine. So here's an example of a tyrosine sulfation site, and each letter represents a different amino acid. And the one that's important for you to remember is Y represents tyrosine and will always be highlighted in red.
So UniProt was the source for all of our amino acid sequences, and UniProt is a protein database with information about amino acid sequence, structure, and topology.
So next we use the position-specific scoring matrix, or PSSM, to score each tyrosine sulfation site. And what it does is it predicts the probability of sulfation based on the amino acid sequences surrounding the tyrosine. What it uses is 102 known sulfated sites and 499 known unsulfated sites. And using this information, it can predict the probability of sulfation at each amino acid surrounding the tyrosine. Here's an example of a scored tyrosine sulfation site with a different score under each amino acid. To calculate the score, you just add the scores of all the amino acids to get a final score. The score of 1 was the benchmark for any tyrosine sites that we looked at because the lowest known sulfated GPCR has a score of 0.92. Then the benchmark for a high score was 2.42 because that is known to be an extremely high score. So this score of 4.54 is really, really high, and the reason it's this high is because of the 3 acidic residues coming right before the tyrosine.
We also use the program muscle alignment in order to look for repetition and conservation among the amino acid sequences. So here's an example of a muscle alignment output comparing the hemoglobin of a goat, a human, and a mouse. And so here we see the amino acid sequences and the letters, and the asterisk notes that at that residue, the amino acid is the same for all 3 species, which is conservation, and not having an asterisk notes that the amino acid at that residue is different for all 3 species, and that would be a substitution.
So next, it was my favorite program, which was PyMol, where we'd go to the protein database and search for a specific protein. And then we'd get a 4-digit code to enter into PyMol, and it would give us a 3D visual of the protein. Then we were able to zoom in on specific residues in tyrosine to actually visualize what was happening around the tyrosine to see what was occurring. And this is an image of apoptosome, which is a protein responsible for programmed cell death. And we put it on our presentation just because we thought it was a very, very pretty protein.
So I study tyrosine sulfation in voltage-gated sodium channels, which are also known as NAV, Na for sodium and V for voltage-gated. Voltage-gated sodium channels are found in various parts of the body, and they commence an action potential along a membrane in order to transmit messages to the brain and regulate heartbeats. So, like I said, voltage-gated sodium channels are found in various parts of the body, and humans have 9 voltage-gated sodium channels known as NAV 1.1 through 1.9. NAV 1.1, 1.2, and 1.3 are found in the central nervous system. NAV 1.4 and 1.5 are found in skeletal tissue and cardiac muscle. NAV 1.6 is found in both the central nervous system and the peripheral nervous system. And NAV 1.7, 1.8, and 1.9 are found in the peripheral nervous system. So here we have a diagram of the structure of a voltage-gated sodium channel. And as you can see, it is broken into 4 domains noted by the Roman numerals. And each domain is split into 6 transmembrane helices. Segments 1 through 4 are responsible for voltage sensing, though 4 is most responsible, noted by the plus signs. Segments 5 and 6 surround the pore and are responsible for gating. And while this diagram looks flat, it actually curves into a circle where the pore is right in the middle and the 4 domains are the 4 on the outside. Here we have a phylogenetic tree of the evolution of voltage-gated sodium channels among each other. Potassium channels were actually the first of the voltage-gated ion family, and sodium channels evolved from calcium channels, a channel that was once permeable to both sodium and calcium before developing their own selectivity filters. So why study tyrosine sulfation in voltage-gated sodium channels? Mutations in voltage-gated sodium channels cause a variety of serious neurological and cardiac disorders. There are many neurotoxin receptor sites in voltage-gated sodium channels, and pharmaceutical drugs for pain and depression are targeted to sodium channels. So why does tyrosine sulfation matter? Finding and understanding tyrosine sulfation sites in sodium channels will help scientists understand and prevent these severe neurological and cardiac disorders, understand the binding of neurotoxin receptor sites, and better target drugs for pain and depression to the sodium channels. So here are my results. 39 tyrosine sites with scores greater than 1 were found. Of those, 22 had scores greater than the benchmark of 2.42. My standard deviation, my average was 2.89, and my standard deviation was 1.46. So here we have where the high-scoring tyrosines were located in the voltage-gated sodium channels. So 82 were located in the pore loop, and 82% were located in the pore loop, and 18% were located in the extracellular segment between S1 and S2. And this means that the high-scoring tyrosines were highly concentrated in only 2 areas of the sodium channel. So on this diagram, the pore loop is right here. Oh, there, it's right there. And the extracellular segment between S1 and S2 is right there. So those are the only locations that the high-scoring tyrosines are concentrated. So the first thing I looked for in predicting my tyrosine sulfation sites in the sodium channels was conservation between the amino acid sequences of the different sodium channels. So here we have the channel, the tyrosine position in the sequence. The sequence and the dots show that the amino acid is the same as the one in NAV 1.1, and the tyrosine score. So here we can see that at conservation site 1, which is defined as position 360 in NAV 1.1, the sequences are very, very highly conserved, with only 2 substitutions in NAV 1.4 and NAV 1.7, and all of the tyrosine scores are greater than the benchmark of 2.42. Conservation site 2 is defined as position 790 in NAV 1.1, and though the conservation isn't quite as high, the amino acids being substituted are similar amino acids and maintain their function, and this conservation site has 4 of the 9 voltage-gated sodium channels. Conservation site 3 is defined as position 1458 in NAV 1.1, and though it is not quite as highly conserved, this conservation site contains 8 of the 9 voltage-gated sodium channels, and all of the tyrosine scores except 1 are greater than the benchmark of 2.42. So this conservation data reflects the evolution we find of the evolution of voltage-gated sodium channels. The conservation data shows that now 1.1, 1.2, and 1.3 are highly conserved, which is why they are also very evolutionarily similar. It also shows that now 1.5, 1.8, and 1.9 are most unique, which is why they are also separate from the other channels in evolution. The only thing not quite explained by my conservation data in the evolution is why NAV 1.4 is separate from NAV 1.1, 1.2, and 1.3 when it was very highly conserved with those channels. So the next thing I looked at to predict my tyrosine sulfation sites was natural variants and mutations. And so natural variants and mutations were found in NAV 1.1 and 1.2. And here we have the channel, the tyrosine position, the sequence, the mutation, which is noted as the new mutant amino acid, the position, and the original amino acid. So for example, in NAV 1.1 at position 790, a tyrosine is substituted by a cysteine, and then we have the tyrosine score and the mutant score. And so the mutant scores were calculated from the PSSM with the mutation instead of the original amino acid. And so all of the mutant scores are much lower than the original tyrosine scores, which suggests tyrosine sulfation because it shows that when the mutation takes place, tyrosine sulfation is less likely to occur. And so some of the mutations do not have mutant scores, and that is because the mutation occurred at the actual tyrosine. And in the PSSM, when at the tyrosine position, the score is always 0, so mutant scores could not be calculated. So tyrosine sulfation is highly likely in conservation sites 1, 2, and 3, and at position 1228 in NAV 1.5, because all of the scores from the PSSM were greater than 1. The sites were well conserved among the different sodium channels. Mutations in near or at the tyrosine sites cause very serious disorders, and the mutant scores were all lower than the original tyrosine scores. So now, what does this mean? So first of all, sodium channels have no signal peptide. What this means is that signal peptides are usually required to reach the Golgi complex, which is where tyrosine sulfation takes place. And sodium channels have no signal peptide. However, they can be glycosylated or sialylated, both processes that occur in the Golgi complex. Which means that while sodium channels have no signal peptide, they can still go through this Golgi complex and be sulfated. So the mutations I found were in NAV 1.1 and 1.5. NAV 1.1 is a neuronal sodium channel, which means that the mutations caused very serious epilepsy disorders. Dravet syndrome is previously known as severe myoclonic epilepsy in infants and occurs in the first year of life. GEFS plus is genetic epilepsy with febrile seizures plus, and also generally happens in childhood, though can endure throughout your life. NAV 1.5 is a cardiac sodium channel and so was associated with severe cardiac disorders. Brugada syndrome is the cause for 20% of all sudden cardiac death. And long QT syndrome is a cardiac disorder characterized by a longer QT interval, which is the interval your heart takes to recharge after a heartbeat. So when that interval is longer, sudden death can occur from exercise or emotional stress. So in conclusion, tyrosine sulfation has a huge role in promoting normal function because of its property of enhancing protein-to-protein interactions. And because of this property, when the tyrosine in a tyrosine site is sulfated, the protein-protein interactions are increased and the mutations are less likely to occur. Tyrosine sulfation also has a huge role in drug charting, similarly because it has the property of enhancing protein-to-protein interactions. And understanding tyrosine sulfation sites will better help scientists specifically target drugs to the tyrosine sulfation sites in sodium channels. I would like to acknowledge Dr. Grace Rosenquist for guiding me through this research experience. I would like to thank Kate Blanchard, my lab partner, for being with me and helping me through along the way. I would like to thank the YSP staff for giving me this amazing research opportunity, and I would like to thank my family and friends for supporting me in my life.
All right, hi everyone, I'm back, I'm Kate Blanchard, and I'm here to talk about Class C G protein coupled receptors. And the reason I studied this specific set of proteins is because chemokine receptor A and the other, or chemokine receptor 5 and the other chemokine receptors are all Class A GPCRs. So we'd expect similar proteins to also have tyrosine sulfation, and therefore we'd expect Class C GPCRs also to have sulfated tyrosines. And currently there are no known Class C sulfated GPCRs. So here's the structure of a GPCR. So it is classified with 7 transmembrane helices, which means that it goes through the membrane 7 times. And what happens is a ligand binds to the GPCR, and if it's an agonist, it will undergo a conformational change. And if it's an antagonist, nothing will happen. So then if an agonist binds to the GPCR, it undergoes a conformational change, and the GDP exchanges for a GTP, which allows the alpha particle to dissociate from the beta and gamma particles. Then the alpha particle can control another protein, thus sending a signal via a second messenger to somewhere else in the body, causing intracellular change. Then the GTP can hydrolyze back to the GDP, allowing the GPCR to reset, ready to bind to another ligand. This is the process that controls virtually all known physiological processes, including tasting and smelling. Humans have over 1,000 known types of GPCRs, therefore they're clearly really important in our lives. So there are 3 different classes of GPCRs, classes A, B, and C. Class A is the class that all the chemokine receptors are. And the reason class C is different than the other classes is because of this large extracellular domain. This is really important for tyrosine sulfation because the tyrosines can only be sulfated if TPST, the catalyst, can reach the tyrosines. And this happens in the extracellular domain. So as you can see here, Class C's domain is much larger than A, so clearly there is a larger area for TPST to reach and therefore more tyrosines to be sulfated, so this is really important for tyrosine sulfation. Some examples of Class C GPCRs are GABA B receptors, calcium sensing receptors, taste receptors, Class C orphans, and metabotropic glutamate receptors. So the reason tyrosine sulfation in GPCRs matters is because over 50% of pharmaceutical drugs are targeted at GPCRs. Therefore, in order to help scientists design the most effective drugs, they need to know the complete structure of GPCRs, and therefore they would like to know which tyrosines are sulfated and how they affect protein-to-protein interaction. So here are my predictions. First, overall scoring. I started with 63 sites that scored above 1.0. Almost 50% of them scored above 2.42, and the average was 2.73. So then I focused on a very specific site that had 12 scores scored above 1. 75% of them were above 2.42, and the average was 3.9. So this is clearly well above 2.42, which was our benchmark for being a very high score. So next we wanted to localize where these tyrosines were, so that's when we checked for conservation. Here's part 1 of the conservation of the focus site, and it just shows all the tyrosines I talk about are going to be in this early 200s region, which will ultimately be really important. But first you see that the first column lists the names of the receptors, and these are all the 8 different metabotropic glutamate receptors. Then the next column you see the position of the tyrosine, and the final column you see the sequence. And similar to Catherine, you see a lot of conservation throughout these different receptors. And where there are substitutions, they're still between similar amino acids with similar properties, like large hydrophobic or polar. Therefore, we expect them to act in a very similar way, so the substitutions aren't as major as they actually appear on the screen. Next, the second half of conservation, also in the early 200 site, is between the calcium sensing receptor, GPRC6A, which is an olfactory or smelling receptor, as well as taste receptors 1, 2, and 3. And you can see further conservation between these receptors, and therefore we can localize all of these tyrosines into this one early 200s area on the N-terminus or in the extracellular domain. Not only was there conservation between receptors within humans, but there was also conservation between different organisms within the same receptors. So on the screen you see an example of metabotropic glutamate receptor 1 comparing humans, mice, zebrafish, and fruit flies. And what you can see is that humans, mice, and zebrafish all have the exact same sequence. And then when you compare humans and fruit flies, there is still a 60% conserved sequence, which is really important because you would never expect humans and fruit flies to have similar sequences because one's a human and one's an insect. But you still see that when you're comparing metabotropic glutamate receptor 1, there's still a very highly conserved sequence, which allows us to really know that the tyrosine in this location is a very important location. Next, moving on to mutations. There were 12 mutations on 6 different receptors, and what happened for all these mutations is there was a loss of protein function and the tyrosine was no longer there. This indicates tyrosine sulfation because the tyrosines have the property of increasing protein-to-protein interaction. So if the tyrosine is no longer there and the new amino acid does not have the same property, you would not expect the protein to function properly, and therefore you would, the protein would be able to function if the new amino acid was there. And here's an example of a mutation for metabotropic glutamate receptor 3. You start with a tyrosine at position 222, and then an alanine replaces the tyrosine, and then the protein loses its function, indicating tyrosine sulfation. And finally, the location of the tyrosines also indicated sulfation. And this is an image from Pymol of the calcium sensing receptor on residue 218 with the tyrosine highlighted in pink. And what you can see is that the tyrosine is literally sticking out of the protein, clearly accessible by TPST, and therefore it would be able to be sulfated, also indicating tyrosine sulfation likely. So when you take all of this evidence into account, with the high PSSM scores, the conservation within species, the conservation across species, mutations causing protein dysfunction, and having tyrosines on the surface of the protein, they all point to yes, the tyrosines are sulfated. So now we need to understand what this actually means in the real world. First, this shows us that the ligand binding site is highly probable to be impacted by sulfation. And the reason we believe this is because the ligand binding site for class C GPCRs is on the long amino terminal domain or on the N-terminus, which is the most important part of the protein. Because in order for a GPCR to undergo a conformational change, it needs to be able to bind to a ligand or else no signal will be sent. And what happens with tyrosine is that evidence has shown sites conserved with tyrosine 236 is that it has a hydrogen bond to glutamate for the metabotropic glutamate receptors. So what this means is that one of the bonds that allows the glutamate to bind to the ligand is actually a tyrosine. So if that tyrosine was sulfated, it shows that tyrosine sulfation has a large impact and importance in the actual function of GPCRs overall. And this is really, really important for tyrosine sulfation because the most important thing is that the GPCR can send a signal and it can't do this without binding to the ligand, and the ligand can't bind without the tyrosine. But next, the effects of mutations. First, for the calcium sensing receptor, with the mutations cause homeostatic disorders or fatal neonatal severe primary hyperparathyroidism. And this is a disease that has an 87.5% mortality rate within the first year of life. But next, we're looking at metabotropic glutamate receptor 2. It abolishes binding for Glumagad, which is a drug that has potential to treat anxiety and drug addiction. And finally, for metabotropic glutamate receptor 3, it abolishes binding for DCG4, which is a ligand with neuroproductive and anticonvulsant effects. So what's really interesting about these mutations is that while one mutation actually causes diseases, two of the other mutations actually cause problems when trying to treat disease. So there are various mutations on all ends of the pharmaceutical spectrum, so they are very important for us to understand how tyrosine sulfation affects it and what actually causes these problems. So in conclusion, we can find that tyrosine sulfation is likely at the predicted ligand binding site, which is extremely important because that is the most important site of the protein. And furthermore, this information gives scientists the ability to better design pharmaceutical drugs, because if they understand how tyrosine sulfation affects ligand binding, they can design their drugs more effectively to bind to these GPCRs and proteins, and therefore enhance a better quality of life for many people. I would like to thank Dr. Grace Rosenquist for guiding me through the researching and paper writing process and teaching me about tyrosine sulfation. And I would like to thank Catherine Jacobs for always being there to support me throughout the paper writing and researching process as well. And I'd like to thank the YSP staff for always being there for us and holding our research group meetings and making sure we were all on track. And I'd like to thank my family and friends for always supporting me throughout my life as well. And here are my references. And now Catherine and I are available for any questions you guys might have.
Yeah. So Catherine, I have a question for you. So you had said that you only found high-scoring tyrosines in 2 different sites on the sodium channel. Do you know why, or like have any predictions for why this occurred?
Yeah, can I go back to that slide actually? Or go back. Okay, right here. So right here, the high-scoring tyrosines were only found in this region, the pore loop between S5 and S6, and the extracellular region between S1 and S2. And this is because, this is why I think the pore loop has the biggest extracellular domain of the sodium channels. Like in every domain, 1, 2, 3, and 4, it has the biggest extracellular domain, and that is where the catalyst TPST can reach. So that's why I think that the pore loop has the most high-scoring tyrosines. And then in the extracellular segment between S1 and S2, I found that a binding site for beta scorpion toxin. However, I did not, in my research, I didn't find that any tyrosines were near or part of that binding site. So in further research, I would hope to find that tyrosine sulfation has something to do with that beta scorpion binding, but as of right now, I don't know. Any other questions?
Yes, Ellie?
Can you clarify the role of tyrosine binding?
Yeah, so basically what happens is that in order for a ligand to bind to a GPCR, it needs to bind to numerous different residues to bind properly. And then what I found in my research is that a tyrosine is actually one of the bonds that is bonded to the ligand. Therefore, it's really important in order for the ligand to bind correctly to have a tyrosine present, and we'd expect those tyrosines to be sulfated, which is why sulfation is important as well. Yes, Stephen?
So in yours, you said that when they have a different amino acid there, that it loses the function of the protein, it stops working. And you said that indicated sulfation.
Yes.
How does that indicate sulfation and not just that it was the amino acid that was substituted?
So what we find is that since tyrosine sulfation increases protein-to-protein interaction, and then since the tyrosines that we were focusing on had high conservation, had high scores, were at the surface of the proteins, we already predicted that they were sulfated. And then the fact that when they weren't there anymore, and if they had that property of increasing protein-to-protein interaction, if it wasn't there and wasn't able to bind and help the protein function properly, we expected that sulfated tyrosine loss caused protein dysfunction. Yeah, John?
So can you explain how changes to the extracellular tyrosines and how those are sulfated will impact the intracellular behavior as it relates to alpha, gamma, beta particles?
Oh, so basically the reason I talked about the alpha, beta, and gamma particles was just to explain how the protein functions, but what the impact of tyrosine sulfation is actually just the ligand binding, because then once the sulfated tyrosines are there and once the ligand can bind properly, it just makes the protein function, because without tyrosine sulfation, without ligands binding, the alpha and beta and gamma particles can't ever actually have any function because there's no ligand to start the process to have the function.
Yes?
I've asked this to Kathy because you've been answering a lot of questions.
Can you explain again why the mutations that aren't at the tyrosine actually matter?
Yeah, so this has to do with sort of how the PSSM scores them, because when we looked at the PSSM, the amino acids surrounding the tyrosine are important for whether the tyrosine is sulfated. So mutations around the tyrosine and that aren't actually at the tyrosine can also be important in understanding whether or not the tyrosine is sulfated.
And also to add on to my answer to John's question about the intracellular function, so we didn't focus on any tyrosines that were actually in the cell because TPST can't sulfate them. So those tyrosines that are sulfated within, like that won't happen. So that's why we only focused on the extracellular tyrosines as well. And next up, we have Caitlin Lim.
Caitlin, is everyone here who's you're expecting to be here?
Yes.
Perfect.
Good morning, everyone. My name is Caitlin Lim, and for the past 6 weeks as part of the Young Scholars Program, I have worked under the direct supervision of Ian Korf in the Korf Lab, and my main project had to deal with software development, where I created a tool called LACOON, which is essentially a tool used for automatic cell counting using images. So, before I get into the actual meat of what the project entailed, I'm going to give you guys a little bit of background first, and that's going to include talking about cell cycles and more specifically how they pertain to retinal cells. So, the cell cycle, I'm sure most, if not all of you are familiar with, details the life of a eukaryotic cell, and as you can see in the figure, it can be divided into 4 main parts. So the first part is G1, or the first growth phase. This is when the cell starts kind of developing. And then next is the S phase, or the DNA replication or DNA synthesis phase. This is when the cell starts kind of its process leading towards mitosis. And then there's the G2 phase, which is further growth and preparation for mitosis. And then lastly, there is M, which is the actual mitosis, or the dividing of the cell. And so how is this specifically pertinent to retinal cells? Well, the cell cycle is especially important for retinal cell development because retinal cells first start off as a blob of undifferentiated stem cells. And then, as you can see in the image, they kind of develop in layers. And so because the retinal cells, or the retina itself, develop in layers, it is important that the cell cycle of these stem cells is regulated so that they know when to differentiate when and how. And one of the factors, or one of the molecules that is especially important for this, for the cell cycle regulation, is using microRNAs. MicroRNAs, or miRNAs, are especially important, they are non-coding, they're small non-coding portions of RNA, and they're important, as you can see, in retinal cell development. And today, we're going to be focusing on one specific microRNA called LET7. And so diseased retinal cells have 2 characteristics, or one of 2 characteristics regarding LET7. So they can either have a lack of LET7 or LET7 knockout, or they can have a lot of LET7, which is LET7 overexpression. And then so past research has shown that diseased retinal cells might have different proportions of cells in certain phases relative to healthy retinal cells. For instance, diseased retinal cells might stay in the S phase for longer compared to healthy retinal cells. And so in order to investigate this, we can then use cell counting. And that leads me to my motivations. Why does any of this matter? Well, generally speaking, cell counting is extremely important in both research and clinical applications. And there is automation that exists in the form of plugins with the application ImageJ, which is a biological imaging software that is available for the open public. And although automation exists, the most recent plugin used for automatic cell counting was last updated in 2010. And so this shows that these softwares are outdated and they're not maintained properly. And so this calls for a new software or a new package that can then count these cells automatically and efficiently using new algorithms that might have developed in the time between. And so now we're going to go into the images that I used, or the testing data that I used to kind of train my software and to create my software. So I have 108 images used in total, and they could be divided into 3 main components. The first is the control. which are images of healthy and normal retinal cells. And then the second is LET7 knockout, or retinal cells with LET7 knockout, so they have dysfunctional development. And the third and the last is retinal cells with LET7 overexpression, so they have accelerated development, but they're still dysfunctional. And all of these images have in place two forms of staining. There is EDU staining. And so EDU is a type of molecule that essentially acts as a nucleotide analog. And so it is injected into the genetic material, or the DNA. And when DNA is replicated, that is when this EDU molecule is expressed. As a result, the fluorescence is expressed during the S phase of the cell cycle. And there is also Fuchi staining, which takes advantage of two different fluorescent molecules, GFP and RFP. And so the RFP is expressed during the G1 phase of the cell cycle, whereas GFP is expressed during S and G2. And so among all of these images, all of the images can belong to one of four different channels. So first there's the DAPI channel, which contains the image of all the cells regardless of what cell cycle phase that they are in. And next we have the EDU channel, which shows images of retinal cells that are in the S phase. And then the RFP channel, which shows cells that are in the G1 phase. And then the GFP channel, which shows cells that are either in S, G2, or F. And this figure just shows a sample of the images that I worked with. And so the upper left shows the blue cells, which are the DAPI channel. The upper right shows cells in the EDU channel, they're black and white. The lower left shows cells in the GFP channel, they're green. And then the lower right shows images of cells in the RFP channel, and they're red. And so now, kind of detailing the process that I took to develop the software, the process can be divided into three main components. First is image training, so actually using the images to create the software. The second is quality control, where I kind of made sure that my code was working to the best of its ability. And then lastly, distribution, making my code accessible to the general public. So first we're going to start off with image training. And I'm going to be detailing a few key algorithms that I used in the process. And so histogram equalization is a sort of pre-processing step. And pre-processing images is it's useful in that after an image is pre-processed, it helps the actual algorithm, the actual cell counting code, better understand or better work with the images. And so, what histogram equalization does is that it essentially enhances the contrast. As you can see in figure 5, the original image can be represented as a histogram, or distribution of pixel brightnesses. And in the transformed image, you can see that the histogram is essentially stretched out. So that is what histogram equalization is, and as a result, you can see the image is contrasted much more. The second step of pre-processing that I've gone under is Gaussian filtering. And as you can see in this figure, the original image is of this guy, and then the Gaussian filter image shows a much more blurred, a much more smoothed image. And this is essentially what Gaussian filtering does. It smoothens the image, and it removes any possible noise that might be present in the image. And now that we've gotten the pre-processing methods over with, now we can actually talk about the actual algorithm that we used in order to count the cells. And so the main algorithm that I used is what is called regmax, which is short for regional maximum. And so this essentially takes an image, a black and white image of cells, and finds the regional brightest points, which it then identifies as the centers of these cells. As you can see in this figure, the left shows the original RFP channel image, while the right shows the detected centers, or the centers that the computer detects after using the regmax function. Next is quality control, because not all code is perfect. So the main form of quality control that I used was something called epsilon values. And because we want to make sure that we're counting actual cells, not just random specks or dust or random pieces of cell debris. And so the underlying motivation behind this algorithm is shown in this figure. You can think of the black blob as kind of the general cell, and then the blue circle as the detected cell center in the DAPI channel, and then the red circle as the detected center in the RFP channel. As you can see, even if they denote the same cell, the centers don't really align. And so what epsilon value counting does is that it uses that epsilon value, which is predetermined, and it kind of uses that as a radius to sweep around its surrounding area to detect any other cell centers in other channels, and it counts that and make sure that they're counting the same cell. And of course, how did I actually implement this in the code, and how accurate is the code in the first place? So, when I was writing the code, I made 4 main methods, or 4 main pipelines of cell counting. And it's important to note that all of these have Gaussian filter preprocessing, and all of these use the regmax function to actually count the cell centers. But the only way that they differ is by whether they add in histogram equalization as an extra step of pre-processing or epsilon values as an extra step of quality control. And so how well did this actually perform? So I calculated the average experimental error compared to the actual counts, which was made by actual researchers. And from the results, I could see that the DAPI counting performance does not change depending on what pipeline that you use. Whereas for RFP and GFP channels, the counting works best when only epsilon value quality control is implemented. Whereas for the EDU counting, it actually works worse with only epsilon value quality control. But the average experimental error is consistent otherwise. And lastly, distribution. I need to make my code accessible to the public so that they can actually use it. So how did I do that? Well, I used GitHub, which is a platform where people can upload code and it can be used by other people. And so this is a screenshot of the README file that kind of details on my GitHub that details how to use this project and how to actually execute the software. Alternatively, you can also just use your command line on a computer. So this GIF shows how you can install the software on your computer by using the command line. As you can see, it says pip install and then the software name. And so you can just type in pip install LOAC1, and then you're done. So, future directions. What else can I do to further improve this project? Well, the underlying motivations behind future directions is to, of course, improve the accuracy of the software, make sure that it's counting the cells perfectly all the time. And so one of the ways that I was thinking about going about with this is to implement a more effective means of quality control, exploring just beyond using epsilon values and seeing what other methods that we can use. And of course, the second, albeit a more ambitious approach, is to create a larger library of cell counting tools so researchers have a wide selection of different tools to choose from, which leads into my third future direction, which is to expand beyond just fluorescently stained cells, so that LOAC1 can be used for any type of cell image. And that basically concludes my presentation. And so first off, I want to thank Ian Korf, my primary research mentor, who I worked with throughout the entire duration of the program. And he gave me a lot of advice on how to kind of improve this project and how to improve the software. I also want to thank Michaela Louie for introducing me to the project in general and kind of talking to me about what this project entails. And of course, I want to thank all the YSP staff and all the counselors for planning all the events and being amazing mentors throughout this 6-week process. And of course, all of you guys. Thank you for making this summer so unforgettable.
So how does your software improve on past software done?
So I actually haven't implemented like comparisons between the ImageJ and the current methods used, but one way that I think can possibly improve is that the current plugins that are used in ImageJ, they're not entirely automatic in that you have to like input a region of interest, you have to input a lot of different values, and so there is still a manual component. Whereas this software package, you just run one script, just one line of code, and then it should be done for you.
When you were analyzing the accuracy of algorithms, why did you use average experimental error rather than specificity and sensitivity?
So the reason why I did that is because if I try to assess the specificity and sensitivity metrics of the model, then I would have to use the training images would actually have to be labeled as whether or not a pixel is part of a cell or not. Unfortunately, because the images that I used were not labeled in that manner, I could not perform those types of metrics.
You use RegMax to detect the cell centers. Are there any other ways to do that in a more efficient manner, or like any other methods?
Yeah, so one other method that might be of further interest is using what are called convolutional neural networks. And so those are just very complex, very refined kind of methods of image segmentation that, although take a lot of time to train and kind of refine, can actually yield more accurate results. And next we have Joy Liu.
Joy, is everyone here for you?
Yeah.
And is anyone watching from home or?
No.
Okay, cool.
Joy, and I'm doing research under the direct supervision of Dr. Ian Korf in the Korf Lab in the UCB's Genome Research Lab. So genomes and their gene parts are the foundation of modern biology. However, what people don't know is that in these foundations, there are cracks. And my research project is essentially to find and fix these cracks. Before we get started, let me tell you about how gene identification works. So, historically, how genes are found is by observing a phenotype, such as sickle cell anemia, and eventually finding the protein or gene that's responsible for it. However, this is very time-consuming and may take weeks to months to even years at a time. Because of that, today we use a more convenient and efficient way, which is that we take the sequence mRNA strand and extrapolate or predict what type of genes and proteins can be coded from. So how do we do that? We do this by using open reading frames, which are essentially candidate genes. Open reading frames are any section of nucleotide sequences that can begin with a start codon and end with a stop codon. Because of that, the mRNA strand can have many open reading frames at a time. Thus, we need a clear method to determine which one is the gene. How we do that right now is we use the longest open reading frame rule. As you can see, essentially what this means is that we take all the potential open reading frames, take the longest one, and just say, okay, that's probably a gene, so let's call that a gene. The reason, it does not sound very scientific, but the reason behind it is because the probability for an open reading frame to be very long is low. So, as you can see from the simulated distribution, the open reading frames beyond 300 nucleotides long is very low to occur just by chance. Because of that, scientists assume that it's got to have a reason for it to be this long, in which it is that it codes for a gene. However, there are certain issues associated with this assumption. For example, the longest open reading frame rule only takes into account the factor of length, and not other factors such as the composition of the sequence itself. For example, if you look at this sequence over here, there are two potential open reading frames. One of them is this whole thing, and the second one is this one, which starts with this start and this end. The longest open reading frame model would find this whole entire sequence as the longest open reading frame and call that a gene. However, if you look, there's a bunch of poly A's in the middle of this open reading frame, so it is not actually biologically likely for it to be a gene. Therefore, biologically speaking, the shorter one may actually be the better alternative, and longer open reading frames does not mean it's always a better fit. Therefore, the goal for our program is to develop and assess if the longest open reading frame rule is creating cracks, and also if there is a better alternative to the longest open reading frame rule to assess which one assess genes in mRNA sequences. In this program, we will be using C. elegans genome because it is the first sequenced multicellular organism, and in addition makes the genome more complete to work with. So here are the methods and how we approach this problem. For an alternative model to the longest ORF model, we use the hidden Markov model, or HMM, which is basically a probabilistic model that is trained to recognize genes much like how facial recognition works. This is, according to our HMM model, what an mRNA strand would look like. As you can see, it has untranslated regions on both ends, and the Kozak consensus, as well as the coding sequence start and stop codon. For our data source, we use worm-based C. elegans one, and we use that to both train and test our data. For our coding source, we use the Grimoire library, which is developed by our team at the Korf Lab. And Grimoire library is basically a biological sequence analysis package, and it can make and test their Markov models. So here are the results that we got from this HMM model. Okay. This is a screenshot of the sequences and genes that I found. So it looks like a lot of words, so let me make it simple for you. Every single section is a gene that's predicted by a model. So all the models with the LORF, they're the genes that are predicted by the longest open reading frame model. And all the ones with M1 and M2 are the genes predicted by the hidden Markov model. As you can see, some of these, the longest open reading frame model is longer, but what this is saying is that it's not necessarily the better one, and these two models are actually more biologically and statistically likely than this model. So how does this look macroscopically? We used that on the entire genome, and we found that 91.3% of the time, those two models predicted the same type of open reading frame as the gene, which is reassuring because that means our entire genome is not completely screwed up. But at the same time, 8.7% of the time, they predicted different results. And what is especially concerning is that 1.1% of the time that they have predicted different stop codons, which means the complete reading frame could be completely off, and then they could be coding for completely different proteins. 1.1% might not seem like a large number, but considering that there are 20,000 genes in a C. elegans genome, that means that 200 of them are wrong. Yeah. Okay. So, how do we know which one is actually correct? What if it's just because our model is not accurate? So we categorized out of all of difference, we put them into categories by using Warm-Base. And all of the red bars, they're the categories where the open reading frame rule, the longest open reading frame rule, is wrong. And all of the green bar is where the longest open reading frame rule is right. And the blue one is where we need more information for indeterminate. As you can see, this is not the complete sample, and we just took it out of a sample of 117 predictions, but 11 out of 117 are correct, and a lot of these are actually incorrect, and we should choose the HMM model instead. Interestingly enough, 14 out of 117 models of the longest open reading frame rule actually predicted too short of a longest open reading frame, which leads me to the next assessment of the data. So obviously when we were doing this, we anticipated a lot of the open reading frames predicted by the longest open reading frame rule when we conducted this experiment, we hypothesized that some of the differences are where the open reading frame should actually be shorter. However, we found 39.7% of the differences actually predicted a longer open reading frame than the longest open reading frame rule, which doesn't make sense because the longest open reading frame rule is supposed to find the longest order, right? But it doesn't. So why is that? That is actually, we theorized that it's actually because the genome is constantly updating and the sequences are not constantly updating. So when they first found the longest open reading frame, they were finding it on incomplete data. For example, if we only have a sequence that is this long, the longest ORF rule would say that this is the longest open reading frame. But in reality, the actual sequence could be this long, and the open reading frame could be over here. So this should lead to another issue that the longest open reading frame will have, which is that if our genomes are constantly updating, then using the longest open reading frame will be running it on incomplete data, which means that there's a high chance that the longest open reading frame rule would be wrong. Yeah. And in addition, some genes that we found in this graph are in the completely wrong frame, which means they code from completely different proteins. And there are also many non-coding RNAs, which means the genes aren't even genes at all. So the next step that we could do is to qualify this data some more. So it would be good if we can identify these incomplete open reading frames and have a program that's like, even though this is the most optimal open reading frame right now, this actual sequence should extend longer. And this is possible with hidden Markov models. Another path that we possibly go down is to identify multiple paths. For example, the longest open reading frame will only takes the longest open reading frame. But what if there are multiple possibilities to be genes through alternative splicing? So this is another way that path that we can go down to make sure our genomes are more accurate. And lastly, what is really important that we should be able to do is to be able to automate the result analysis, which is because when we were categorizing all of the difference in comparison, we were doing it manually. So if we could have a program that can do it like automatically, we would be able to assess a much larger database and have a more accurate way of looking at all the other genomes and be able to suggest corrections. Yeah. I would like to thank everyone in the UC Davis Core Lab in the Genome Center, including Dr. Ian Ford and Dr. Hannah Lyman and Dr. Keith Braga, and also everyone at the UC Davis Young Scholars Program, including all the counselors and my friends, for being really supportive of me during this entire program and helping me with this. Thank you.
Of the 1.1% of stop data, how many of those were characterized by sequences that had the same start between the models but a different stop, and how many of them are characterized by ones that had both different stops and different starts?
So out of all of the ones, because different stop codon is more important than different start codon, if they have a different stop codon, we just characterized it as different stops. And I think for most of them, the stops, we didn't really examine data that closely, but for most of them is that they only have a different stop.
You have the percentages of how the C elegans over reading frame like the same, like how the cracks in the genome there. Can you have those percentages translate to humans and their genome? Those numbers?
The issue is that the C. elegans genomes and the human genomes are very different from each other, so we can't clearly extrapolate what the human genome would look like based on those percentages. However, we can run our program with the human genome and find out the percentages.
So let's say if the long work model produces a wrong gene, does that mean that scientists would spend their entire lives studying the wrong gene?
Not their entire lives, but yes. Well, it's a possibility, but that's one of the reasons we are doing this, is to prevent wasted efforts on genes that are not even actual genes. For example, there are genes in the database that are used for medical experimentation. They are not actually genes, and the purpose of this program is basically to eliminate that, so we don't have to waste effort on genes that are actually not. Any other questions?
So when you All right, ladies and gentlemen, thank you. We're back on break. All right, next up we have Aksha Hailajan.
Hi everyone, my name is Aksha Hailajan and I'm a rising junior from Charlotte, North Carolina. For the past 6 weeks, I've been working in the Department of Animal Science under Dr. Russ Hovey and Carmen Banks. My study investigates the effect of prolactin on calcium regulatory gene expression within porcine kidneys. Let's start off by talking about calcium. Calcium is an essential mineral that is required for several physiological processes in the body, like helping our hearts beat and our muscles contract. However, its most important job is keeping our bones strong. Calcium starts accumulating in our bodies right from the beginning, when we're just fetuses. In fact, calcium is most imperative during this time, as fetuses are expected to have 98% of their fetal skeletons already built by birth. This need for calcium also continues after birth, where newborns seek more calcium from their mother's milk to support their rapid bone growth and development. As levels of calcium and the demand for calcium dramatically increase, mothers need to maintain calcium homeostasis in their bodies. The kidney is an important organ for maintaining calcium homeostasis in the body. It does this through a process called calcium reabsorption, which is essentially when calcium is absorbed back into the blood. Calcium reabsorption occurs with the help of multiple proteins that are encoded by the following genes within kidney epithelial cells. This is an epithelial cell that lines the surface of the kidney. The apical membrane is the side of the cell that faces the inside of the kidney, while the basolateral membrane is the side of the cell that faces the outside of the kidney, or the bloodstream. During calcium reabsorption, several genes work together to cycle calcium out of the kidney and into the bloodstream. The TRPV5 gene encodes for a protein that acts as a channel on the apical membrane, bringing calcium inside epithelial cells. After calcium enters the cells, CalB1 or calbindin transports calcium from the apical membrane to the basolateral membrane. Then sodium-calcium exchanger SLC8A1 and plasma membrane calcium ATPase ATP2B1 act as exchangers and exporters that transport calcium from the basolateral membrane into the bloodstream. This is an example of active transport calcium reabsorption. There are also other forms of calcium transport, like passive transport, that include different calcium regulatory genes. This includes CLDN2, which encodes for a tight junction membrane protein called claudin-2, which acts as a pore and allows the flow of calcium ions between epithelial cells. In addition, SLC12A3 encodes for a protein that acts as a sodium chloride cotransporter and facilitates the exchange of calcium and sodium ions and promotes calcium reabsorption. For pigs, there is a struggle to maintain calcium homeostasis. Since pigs need to produce larger and heavier litter sizes, they also need to produce higher amounts of calcium to support their enhanced milk production. This requires a stable calcium homeostasis in their bodies. However, in cases where there isn't enough calcium and homeostasis can't be maintained, pigs experience difficulty birthing and further health complications. Dairy cows also experience similar problems, but pronounced level. They suffer from calcium deficiency or hypocalcemia. This is also known as milk fever, which is a life-threatening disease that can cause stillborn calves and sudden maternal deaths. This is where research about prolactin comes into play. Prolactin is a hormone that is important in mammary gland development and lactation. Since prolactin's levels rise during gestation or pregnancy and lactation, a lot of research has been done on its potential connection to maintaining calcium homeostasis in the body. Past research has focused on prolactin's role in the small intestine and bone. Results from this research has shown an increase in intestinal calcium reabsorption and bone reabsorption, as well as an increase in calcium regulatory gene expression in the intestine. However, there isn't much research being done on prolactin's role within the kidney. Therefore, what I did was I examined the effect of prolactin on calcium regulation in the kidney. I hypothesized that prolactin would increase the gene expression of all of the following genes in porcine kidney explants. For my project, I used kidney explants that were from parapubescent eels that were slaughtered at the UC Davis slaughterhouse. There were 9 samples in total, 3 of which were day 0 samples that were not cultured with anything, as a way of seeing whether there was any gene expression present before any sort of treatment. The next 3 samples were cultured in a serum-free cell culture media, and as a control, is also known as ITS. The last 3 samples were cultured in serum-free cell culture media and 200 nanograms of porcine prolactin to examine the effect of prolactin on calcium regulation. This entire process of tissue examination and RNA extraction was completed prior to my arrival by my mentor, Mark Biggs. I used cDNA that was generated from RNA through reverse transcription. To amplify my target DNA, I then created a mixture of Go-Green Master Mix, water, cDNA from kidney explants, and forward and reverse primers of a selected gene. I then placed these mixtures into a thermocycler, which allowed for the PCR reaction to occur. Following this, I loaded my PCR products onto a 2% agarose gel and ran it at 90 volts for an hour. From this gel electrophoresis, I was able to compare levels of gene expression between 3 different sample groups. Using a pixel density program, I was also able to measure band intensity. This program essentially counted the number of pixels that were in each DNA band, and I was able to use that to compare the 3 sample groups. I also normalized my data to the values of 18S housekeeping gene. This is my first note for my gene SLC12A3. So I used a 1 kb plus ladder to measure the sizes of the DNA strands and make sure that the sizes of the PCR products were correct. I also used a -RT and a water control to check for any amplification of any genomic DNA, and also to check for any other contamination. So as you can tell, that there's high gene expression in my prolactin samples, no gene expression in day 0 samples, and slight gene expression in ITS samples. From my graph, you can tell that there's an increase in gene expression in prolactin-treated samples when compared to ITS samples and day 0 samples. For my gene Claudin2, or CLDN2, there seems to be more consistency in gene expression across all the samples except for sample 8. From my graph, you can see that there's a slight increase in prolactin-treated samples when compared to both day 0 and ITS samples. However, there is more consistency in the band intensities of all 3 sample groups. For my ATP2B1 gene, I had higher, very strong gene expression in my prolactin samples, no gene expression in day 0 samples, and faint gene expression in ITS samples. You can see this in my graph, as there is a dramatic increase on average in prolactin-treated samples when compared to ITS samples and day 0 samples. For my CALB1 gene, I had high gene expression day 0 samples and no gene expression in ITS or prolactin-treated samples. This suggested that I might have had a turn-off in gene expression, and that made this gene inconclusive. I wasn't able to tell whether it's an increase or a decrease. For my next gene, SOC8A1, there was high gene expression in day 0 samples and decreasing gene expression in ITS and prolactin samples. For my first day 0 samples, I had incredibly high gene expression, and there was a slight decrease between ITS and prolactin samples. For my TRPV5 gene, there was a little bit of conflict as I ran 2 PCRs and I experienced a problem where both of my gel results were completely different. So my first gel showed some inconsistencies in gene expression across all of the sample groups. I did have higher gene expression in my prolactin-treated samples when compared to ITS and day 0 samples. In my second gel, I had more consistency in gene expression in all 9 of my samples, and I did see the same increase in gene expression in prolactin-treated samples when compared to ITS and day 0 samples. There was also more consistency between the day 0 and ITS samples, which allowed me to see this increase. In conclusion, my hypothesis was proven partially correct, as not all of the genes presented an increase in calcium regulatory gene expression under the effect of prolactin. Overall, you can see that the genes that presented an increase in gene expression were TRPV5, SLC12A3, and ATP2B1. Claudin2 or CLDN2 presented a slight increase in gene expression, and SLC8A1 experienced a slight decrease in gene expression. CalB1 was inconclusive due to the turnoff of gene expression. After I leave, my mentor Carmen Banks will be continuing this research by completing a quantitative PCR and doing statistical analysis based on her results. While my project kind of focused more on pigs and cows, my research has implications for all mammals, including humans. By continuing to try and understand the role of prolactin within the body, we can come up with solutions for mammals that are suffering from calcium-related diseases. I would like to acknowledge my Dr. Russ Hovey and my mentor Carmen Banks for helping me guiding me throughout this entire program and teaching me every single day. I would also like to thank the rest of the Hovey lab for supporting me in the lab and always teaching me different lab techniques, and I learned a lot from this entire experience. And also thank you to the UC Davis Young Scholars Program for giving me this opportunity to do research in a field that I love. These are my references. Questions?
So going back to the methods of culturing, so why specifically did you choose to use 200 nanograms of fluorescent films?
Right, okay, so, no, I think this is here. Yeah, okay. So I used 200 nanograms of fluorescent prolactin to kind of mimic or simulate the levels of prolactin during pregnancy and lactation in mammals. And also normal levels of prolactin are around 5 nanograms of prolactin, which shows that dramatic increase in the amount of prolactin. Emma?
In the gel electrophoresis process, you said you normalized genes to, using normalized data to a housekeeping gene. What exactly is
Right. So a housekeeping gene essentially shows a constant gene expression no matter what cells or conditions are used. So what it was kind of a reference to check that all of the conditions didn't affect, any of the conditions didn't affect the expression of any of my genes. So it was kind of like a normalizer like I said.
So you talked about different calcium homeostatic disorders between pigs and cows. Is there anything different about the way it affects these animals?
Yeah, so for cows especially, it hits them harder because they suffer from a calcium-related disease called milk fever, which I mentioned before. They can actually die from this disease, as when they're suffering from milk fever, essentially what happens is all of the calcium is rushing out of their body as they need to lactate for their calves and also have calcium for themselves. So this results in a lack of calcium in their bodies, and their hearts progressively slow down, and their muscles get lazier, and they become more lethargic, and eventually they can die if they're not treated.
been studying the regulation of the circadian clock in sunflower disk development. I've been working in the Harvard Lab, which is in the Department of Life Sciences, under direct supervision of Ray Marshall. So the circadian clock is an endogenous biological clock that regulates physiological and biochemical processes. Circadian rhythms from the circadian clock have a periodicity of 24 hours, and essentially the circadian clock tells us when to sleep, when to eat, and when to wake up. In plants like the Arabidopsis circadian clock, this consists of a network of proteins. As you can see right there, these proteins interact in very complicated feedback loops, where they repress and activate each other throughout the day, which results in rhythmic gene expression. So some sunflower anatomy. Sunflowers are not just one flower, they are composite flowers, meaning they have a capitulum or a head, and this capitulum consists of a bundle of disc florets, with ray florets on the outskirts, as you can see right there. And the reason why I'm studying these disc florets over those ray florets on the outskirts is because these disc florets are actually involved in pollination, while these ray florets are male, sterile, and asymmetric, which also makes it harder to quantify them. So protandrous and thesis in sunflowers. So during pollination, sunflowers display protandry, which is when the male reproductive organ, which is the anther, is displayed before the style, which is the female reproductive organ. And this process overall occurs cross-pollination, which increases genetic diversity. And in general, increasing genetic diversity increases your chances of conferring resistance to fungi and bacteria. And thesis in sunflowers happens gradually in whorls, which are centripetal sequences of florets. So right here I have anthesis at a photoperiod of 16 hours light and 8 hours darkness, which I'll be referring to as LD16-8. And as you can see, at around 4 to 6 hours, the anthers have elongated, which is indicated by the red arrow. And then at around 24 to 26 hours after the experiment has started, you can see that the styles have fully elongated, indicated by the presence of the stigmas opening up right there. So, the properties of circadian regulation. The first property is temperature compensation, which says the circadian clock should stay constant in a range of ambient temperatures. And this goes against the general rule of chemistry that says enzymatic processes should happen faster in higher temperatures. The second property of circadian regulation, which is the most relevant property pertaining to my research, is entrainment, which is the clock's ability to synchronize with environmental cues like temperature and light. An example of entrainment is jet lag. So when we travel to different places, we experience different photo periods and different temperatures, and jet lag is a result of our circadian clock adjusting to that. And the last property of circadian regulation is self-sustainability, which is the clock's constancy in the absence of environmental cues. So I'll be moving on to my research question. So I'm trying to figure out whether these disc florets are regulated by the circadian clock or cued by the environment, or if it's a combination of both. So first of all, sunflowers were grown in the growth chamber where they experience LD16-8, and this is when the anthers usually elongate and where the styles usually elongate as well. And then after they matured up to a certain point where we saw the blue, they were taken to an experimental chamber where they underwent different thermal cycles and different photoperiods. In the experimental chambers, we had a Raspberry Pi computer used to collect data and also an infrared light to help us see these sunflowers in these dark conditions. We looked at the individual heads of the sunflowers, and we also looked at the individual disc florets in agar plates. This is me in the growth chambers, and this is our sunflower head setup right here. You can see the infrared cameras, and you can see the Raspberry Pi computer right there. And then right here is our disc floret agar setup. So before I go into my experiments, I want to preface it with a past experiment by Lovello, who is also a plant biologist. So as you can see, this is in a photoperiod of constant white light. The anthers do not elongate at all, and the styles do elongate, but however, they elongate at a decreased rate. And I'll be referring to this as constant light. So my first experiment is putting these in constant light at 16 hours of 30 degrees Celsius and 8 hours of 18 degrees Celsius. This is supposed to simulate day and night temperatures instead of simulating day and night through light and darkness. I'm simulating them through day and night periods. And what I'm expecting to see is anther elongation. Hopefully these are stimulated by temperatures and they think it's day and night. And then my second experiment is light and dark cycles, 12 hours each at 25 degrees Celsius. I'm going to be comparing the results from this with past results from a postdoc in the Harvard lab to see if these are environmentally cued or not. And then my second experiment, like I said, in constant white light, these anthers are inhibited, so they do not elongate. And my second experiment is to try to see how we can rescue these anthers through darkness. So Labello also found that a period of 3 hours of darkness was enough to rescue these anthers. So I'm placing these in constant light conditions where they will be inhibited, and then implementing a 3-hour darkness period at ZT12 to 15, which is 12 hours after the experiment started. And this is just the diagram of the photo period. And then my last experiment is anther rescue through methyl jasmonate. We are placing methyl jasmonate in the agar plates of the dischlorets, and we're growing them in constant light conditions and L12, D12 at different concentrations, both with a control group. And this is what the methyl jasmonate plates look like. So my results. For the individual sunflower heads and the dischlorette plates, we took time courses of them, and then we converted them into a virtual stack image in image J, where they were manually scored by Karinami, and we graded them based on the criteria of Corella swelling, anther elongation, and stout elongation, which usually happens in that order in the LD16-8. And I didn't include this yet, but this is what the Corollas look like. So Corella swelling is when the flowers covering the anthers swell. And then we graded the plates based on the length in image J as well. So moving on to the actual results. So right here, I have a constant light 25 degrees Celsius with the hours dark at ZT12 to 15, and my LD12, 12 at 25 degrees Celsius. It's a bit hard to see, but because the image scaling is a bit off, which is my fault, you can see that these patterns are roughly the same thing. So if you look down here, this happens around, let's say, ZT72, which is 72 hours after we started. If you look at ZT around 72 as well, or like it roughly, they mimic each other. So this tells us that 3 hours of darkness is able to induce anther elongation, which tells us the circadian clock is being entrained by darkness. And it tells us it's being entrained by the start of darkness, not the amount of darkness, as they both receive darkness at the same times, but different amounts of darkness. And if you look right here, you can see the rhythmicities of the growth patterns also indicate circadian regulation, because it's around 24 hours. Another indication of circadian regulation right here is there's a big delay because they were grown in 16.8 and then moved to constant light ZT12 to 15. These are now, what we think right here is that the circadian clock is resetting itself and being entrained by the environment. And our data comparison, I compared it to Nikki Crew, who is a postdoctoral researcher in the Harman lab a long time ago, and we looked at her LD 25 degrees Celsius 16.8, and my constant light 30 degrees Celsius 16, and then 18 degrees at 8 hours. And like I said, Corella swelling happens before anther elongation, so we can infer that Corella swelling happens around here in Nikki's data, which is not reported. And if we imagine Corella swelling happening here, we can see that Corella swelling happens around the same time. Again, it's a bit hard because the image scaling is off. And since they are happening at the same time, this tells us that temperature does stimulate day and night in the corollas, which might hint environmental cues regulating discrete development. However, anthers still do not elongate, so they do not think it's day and night because of the temperatures. And then I'm comparing my LD 25 Celsius 1212 to my LD 25 16.8, and you can see right here. Actually, before I start, just the photo periods in general, these have to wait 16 hours before getting darkness, while these have to wait 12 hours before getting darkness. So in LD 25-12, dark comes earlier. And since we moved their environmental cue earlier, and their response was also shifted along with the environmental cue, we can say that anther elongation is also cued by the environment. Okay, so right here we have the methyl jasmonate plates at different concentrations. Right here we have the control group at 0 micromolar, and right here we can see most of them elongated for the most part, except this one thing. And then looking at the 100 micromolar, you can see that the anthers did not elongate at all. And this is also constant in the constant light conditions, but we only have the LD 1212 because the constant light conditions, they already inhibit anther elongation, so it's kind of inconclusive, so we can't really determine whether methyl jasmonate has inhibitory effects or not, or if it's just the constant light. So we can conclude from the LD 1212 that methyl jasmonate does have clear inhibitory effects. So we found out that corollas display similar patterns in LD 16.8 and 25 degrees Celsius, and compared to 30 degrees Celsius at 16 hours and 18 degrees Celsius at 8 hours. This shows us that they're cued by the environment, even though anther elongation is inhibited, but that's kind of expected in constant light. And then we compared the LD 1212 and constant light 3 hours period of darkness, ET 1215. They had roughly the same patterns because, first of all, they received light at 12 hours first, and then followed by a dark period. The dark periods were different, and this shows us that entrainment is not by the amount of darkness, but more like the start of darkness. And then we compared LD 1212 to LD 16.8, which shows us the shift in response with our shift in environmental cues. And lastly, our last piece of evidence for our conclusion is the delay in ZT 12 to 15 right here. We think it's the circadian clock setting. And also the rhythmicity of 24 hours suggests regulation. So, all in all, some of our data shows environmental cues regulating disc-like development, but some of our data also shows circadian regulation. So, what's wrong with this? Now, no one ever said that it's mutually exclusive, so we can conclude that disc-like development is cued by the environment and the circadian clock, because again, they're not mutually exclusive. So, characterizing pathways in circadian biology and just learning about them in general leads to the development of mathematical models that increase crop yield under suboptimal pollinators, and it leads to the development of lines more attractive to pollinators as well. These are my references, and right here are my acknowledgements. I want to make a huge acknowledgement to Corinne Marshall. She's very supportive, and she mentored me throughout the whole program. Very impressive. I want to thank the UC Davis Young Development Program for giving me this opportunity. I want to thank Stacey Parmer for letting me work in her lab. I want to thank the Parmer Lab for helping me with any questions I may have had. I want to thank the YSP counselors for listening to me present this multiple times. And I want to thank all of you guys for making my experience wonderful. Are there any questions? Yes.
So you mentioned earlier that transport and train are
Yeah.
and that's in regard to presence of darkness rather than.
Yeah, so you can see that. Oh, sorry. I'll let you say your question first. Sorry.
So I was wondering, like, is there particular reasons why that is?
We actually don't know right now because circadian biology, the more we learn about it, the more we learn that we don't know about it because so to answer your question, we don't know. Are there any more questions? Yes, Daniel?
In your discussion, you said that there's potential for developing more attractive lines for pollinators. Can you clarify what that means?
So just in general, you can select certain disc florets with different floret depths, and these floret depths can be more attractive to pollinators.
everyone. My name is Emma Xu, and for the past 6 weeks at the Young Scholars Program, I have been characterizing California Agrobacterium tumefaciens walnut isolates in a Kukla lab under the direct supervision of Dr. Ollie McLean from the UC Davis Department of Plant Pathology and USDA Agricultural Research Services. So, what exactly is Agrobacterium tumefaciens? Agrobacterium tumefaciens is a species of bacteria that is known to cause disease in plants called crown gall disease. Agrobacterium tumefaciens is also a natural, opportunistic genetic engineer in that when a plant is wounded, the plant secretes sugars and hormones into its environment, and the species of bacteria is able to pick up on these signals and move towards the plant. Upon interaction with the plant, the bacteria injects tumorigenic DNA and genes into the plant, and this DNA eventually gets integrated into the plant's genome. This integration is expressed through the plant's inability to regulate cell differentiation, resulting in a tumor-like growth at the base of the plant called which is called a gall. This gall produces opiates for this bacterium to thrive on. So why study crown gall disease? Well, walnut species in California are very susceptible to crown gall disease, and California's walnut industry is valued at approximately $1.6 billion a year. And as you can see, a gall in proportion to a tree can have quite a detrimental effect on fruit production. So, in characterizing different Agrobacterium tumefaciens strains, we hypothesized that not only would these strains be very genetically diverse, but they would also exhibit very different phenotypic traits. In carrying out this experiment, I worked with 30 different Agrobacterium tumefaciens isolates from all over California, and these strains were collected from California's top 10 walnut producing counties, including Stanislaus, Yolo County, and more. So in phenotypically characterizing all these different Agrobacterium strains, I performed 3 different tests. The first of which is carbon source utilization. And this is important as it indicates how competitive and how efficient specific strains are at using different carbon sources. The second test I performed was motility, and this is important as it provides insights to how virulent different strains are. And the last test I performed was antibiotic resistance or agrocin resistance tests. And this is indicative of how effective, how competitive certain strains are and how good they are at surviving in their environments. So in carrying out my first test, which is carbon source utilization, I used a product called Biolog ecoplates. And Biolog ecoplates are very effective in conveying data of how good certain microbial communities are at metabolizing and utilizing carbon substrates. And in the test run, using the string ST001, we determined that the 4 carbon sources that had the most significant absorbance changes were D-mannitol, glucose 1-phosphate, alpha-D-lactose, and D-galactonic acid gamma-lactose. So these were the 4 carbon substrates that we mostly focused on. As you can see, this is a Biolog ecoplate, and it is divided into 3 different sections. Each section has 31 carbon substrates and 1 water blank. So with 3 replicates of each carbon substrate, we had more accurate data to analyze. So this is how all 30 different strains of Agrobacterium tumefaciens utilize glucose 1-phosphate. As you can see, most of the strains were pretty good at using glucose 1-phosphate, with some exceptions of outliers, some being particularly inefficient and some being particularly efficient in using glucose 1-phosphate. And these outliers were pretty much consistent throughout the other 3 carbon sources that I analyzed. So these outlier strains, we picked out these outlier strains for further tests. And as you can see, here are the control strains that we picked out, which are ST001, ST003. And these are the inefficient strains, while these were the most efficient strains. So after identifying these strains, we performed motility tests and antibiotic resistance tests. So motility on Stonier's medium. Stonier's medium is a very nutrient deficient medium, and it is only natural that when we spot bacteria in the middle of this medium, that this bacteria expands or grows in search of more nutrients. As you can see, as they grow, a growth halo forms, or this cloud-like structure. So We spotted these bacteria on this medium and incubated it for approximately 48 hours, taking growth halo readings at 24 hours and 48 hours. And this is the data for, this is the growth halo, these are the growth halo readings for 48 hours. As you can see, there's large range at which the growth halos were sized. Some being as small as 0.73 centimeters and others being as big as 2.17 centimeters. The next test that we performed on these 8 different outlier strains was agrocin resistance. Agrobacterium radiobacter is a species of bacteria that produces agrocin, which is an antibiotic that is specific to Agrobacterium tumefaciens. And A. radiobacter produces A. radiobacter is seen as K1026, and we isolated K1026 from a commercial product called Nogal. And after we isolated it, we verified its authenticity through PCR and gel electrophoresis. We then cultured this K1026 and spotted it on Sonier's medium, allowing it to grow for a few days. After a few days, we lysed it with chloroform, and then we poured a suspension of agarose and bacteria over this medium. And after incubating it for a few days, if a particular strain was resistant or was sensitive to agrocin, we were able to observe inhibition zones, or this is where K1026 is effective and bacteria does not grow within that radius. Out of the 8 strains, 5 of the strains were sensitive and 3 of them were resistant. So this shows how the product Nogal that many farmers use before they plant walnut plants may not be the most effective way of controlling Agrobacterium tumefaciens in the field. So what does this tell us? The variance between all these 3 different tests and the different results tell us, it proves our hypothesis correct that these strains are not only genetically diverse, but they also have very different phenotypic traits. And the diversity in these results tell us that Agrobacterium tumefaciens strains most likely did not originate from a single progenitor. As we are still in the process of characterizing various Agrobacterium strains, there are many other characteristic tests that can be implemented. And one that would be particularly interesting to observe would be how different strains interact with each other, as this would be indicative of how competitive and how efficient certain strains are at utilizing food sources. And if in the near future we find that certain Agrobacterium strains from specific areas or counties share certain characteristics, regional solutions to crown gall disease may be developed. And with further research, of course, comes unanswered questions. And one of the big questions that we will look into is, to what extent do these different characteristics, such as carbon source utilization, mobility, or antibiotic resistance, have on the severity of virulence of these specific strains? And another question is, what qualities, or which qualities, or what combination of these qualities make a strain the most competitive and most fit it can be? And another field of interest, another thing to understand for Agrobacterium tumefaciens is how different strains utilize their secretion systems. Secretion systems are the mechanisms that this bacteria uses to inject its tumorigenic genes and DNA into humans. And I would like to acknowledge the Kluppel Lab for fostering such a warm and welcoming environment and teaching me so much for these past 6 weeks. I would like to thank the YSP counselors and the YSP program for making these 6 weeks so enjoyable and fun. And I would like to thank my family at home I'd like to thank my family for supporting me in everything I do. And I want to thank all of you guys for making this past 6 weeks super fun and super memorable. References? Questions? Yes, so crown gall disease is known to have one of the widest host ranges among many plant pathogens. It can actually infect any dicot organisms.
So how did you pick your control strains for covering analysis besides the facility and antibiotic resistance?
Okay, so we picked STA001 and STA003. STA001 is a strain that has been worked with a lot in this lab and it has been characterized a lot, so we started off with that one. And then we picked STA003 because it's from the same county as STA001. And we want to see if there were any correlations noticeable similarities and characteristics between these two specific strains.
You mentioned regional similarities amongst the controls. Were there regional similarities amongst the outliers as well?
Not that we observed. That will be something that further research will be able to identify.
Show us how the tumors look on specific walnut plants, and but you also said it affects other plants. So does the tumor look any different based on?
It pretty much looks the same, but yeah, it's just a